The central dogma of molecular biology is a theory that states information flow is unidirectional. It suggests that information flows from DNA, to messenger RNA, mRNA, to proteins. DNA contains the genetic instructions that are necessary for the synthesis of proteins and other biological molecules. mRNA transcribes the gene sequence from DNA into the amino acid sequence of proteins. Proteins build and maintain structures within an organism, as well as regulate biochemical processes. This flow of information is possible thanks to various biomolecules or enzymes. Proteins synthesized from mRNA are known as transcripts, while the DNA from which it is transcribed is known as the template. Transcription is the process of copying DNA into RNA, and the mRNA type used is mRNA. The mRNA carries the genetic information which is translated by ribosomes into a sequence of amino acids. These amino acids create a protein molecule, which can be used for various functions in the organism. DNA does not codes for proteins directly, but for the mRNA which codes for them. The mRNA is translated into a sequence of amino acids which forms the primary structure of the protein. The translation mechanism is made possible by the different elements involved in the process. These elements include transfer RNA, ribosomal RNA, DNA polymerase, and amino acyltransferase synthetases, among others. The amino acyltransferase synthetases are responsible for recognizing the codons of certain amino acids and joining them with the correct tRNA. Once the translated mRNA molecule leaves the ribosome, it becomes the primary structure of the protein, which is then further modified into its secondary structure. After that, the tertiary structure of the protein is formed, which is responsible for its physical properties and functions. The central dogma of molecular biology is key in understanding the processes of gene expression and regulation. It helps us understand how genetic information is passed on to proteins, and how those proteins are then used in the cell's various physiological processes. Through this process, the genetic material, DNA, is transformed into proteins which govern all biological cells. The central dogma also explains the mechanisms responsible for the heredity and evolution of living organisms. The flow of information is accomplished through a complex network of interrelated proteins, which is essential for the development and functioning of the genetic code. The central dogma is the cornerstone of molecular biology, as it provides a useful framework for understanding gene expression. The architecture of the central dogma has been greatly simplified by the elucidation of the genetic code and its associated mechanisms. Moreover, it underlies much of our current understanding of how the genetic material is organized and regulated.